Dr. Christopher Walsh is, like many Harvard professors, has many, many titles and honors and everything else. So he is the Bullard Professor of Pediatrics and Neurology at Harvard Medical School, the Chief of the Division of Genetics and Genomics at Boston Children's Hospital, an investigator of the Howard Hughes Medical Institute, and an associate member of the Broad Institute as well. He completed his MD and PhD degrees at the University of Chicago, neurology residency and chief residency at Massachusetts General, and postdoctoral training in genetics at the Harvard Medical School. In 1993, he became assistant professor of neurology at Harvard and Beth Israel Deaconess Medical Center, and he has been the Bullard Professor since 1999. He moved to Boston Children's Hospital in 2006, where the Division of Genetics and Genomics diagnoses and treats children with diverse, rare, and orphan diseases, is undertaking more than a dozen clinical trials of innovative medical and gene therapies for genetic disorders. His research has focused on the development, function, and evolution of the human cerebral cortex, pioneering the analysis of genetic diseases that affect the developing brain by fostering worldwide collaborations with physicians and families. His lab has identified genetic causes for dozens of brain diseases of children associated with autism, intellectual disability, seizures, and cerebral palsy. He has discovered that some of these disease genes were important targets of the evolutionary processes that shaped the human brain. In 2017, he inaugurated the Allen Discovery Center for Human Brain Evolution at Boston Children's Hospital and Harvard Medical School, collaboratively with doctors Michael Greenberg and David Reich. And of course, we are going to have David Reich speak in a few weeks as part of this series as well. And this center brings together brain science with evolutionary genetics to search for the key changes in the genome that endow humans with our unique abilities for language, art, culture, and science. He has, of course, been recognized by research awards from many different places that I'm not going to list because really you want me to get off the stage and for him to get on the stage. But I would like to mention that he is an elected member of the American Association of Physicians, the National Academy of Medicine, the American Association for the Advancement of Sciences, the American Academy of Arts and Sciences, and the National Academy of Sciences. So I feel like phew, and we're really looking forward to hearing you speak. Thank you.
Well, thank you so much, Jane, for inviting me to come speak here. I'm really thrilled to come over and talk to a general audience about a lot of the enthusiasm that we have about trying to understand those genetic changes that make us human. And I really appreciate your generous introduction and the fact that you did it in front of one or two members of my family. But anyway, so I'm delighted to have a chance to chat with you all for the next 45 minutes or so about at least how we think about the evolution of the human brain. I'd like to introduce this Allen Discovery Center for Human Brain Evolution. As Jane mentioned, it's a collaboration between our lab and David Reich and Michael Greenberg's lab at the Department of Neurobiology, and also Alice Lee and Ryan Doan. And we'd be nowhere without Soha Ashrafi, who's our program manager. You can visit our website to learn more about the center. And I'll be telling you in the second half of the talk a little bit about what we're trying to do with the center in terms of some of the science programs. So I'm trained, as Jane said, as a developmental neuroscientist in the lab, and then as a neurologist. I never really thought I would be an evolutionary biologist, but you'll get a sense of how I stumbled into that field over the course of the talk, I hope. So I'll just start out by saying none of us see the world the way it really is. What our view of the world is a creation of our sense organs. And every species sees a completely different world. They perceive a completely different world. And you can illustrate that fact by thinking about how a shark sees the world. So a shark hears its prey almost a mile away, and it smells its prey more than 100 meters away. And it can actually feel its prey up to 100 meters away. I have no idea what that would feel like, but for the shark, they have sensory organs in the outside of their skin. And so the sense of touch for a shark is a sense that acts at a distance. It can actually perceive things that are, again, far enough away that we can't even see them in some cases. And then they can perceive actually the electrical activity of its prey up to 50 centimeters away through the water. And I have no idea what that would even feel like, to be able to perceive the electrical activity of someone else who's in this room. And then actually, a shark can taste its prey before it even gets it into its mouth, because it has taste buds all over the outside of its face. And then the shark has such terrible vision that it only finally sees its prey as it's going into its mouth. And so this is a completely different world in terms of trying to picture yourself as a shark, thinking about this blind thing where you're just responding to senses or odors or sensation at a huge distance. So every species is like this. Every species has its own set of sensors, its own sort of brain, and perceives a completely different world. And in fact, of course, even different humans perceive a different world. We know that some people are colorblind and don't perceive the same colors that we do. We know that many animals don't see color at all, similar to some humans. We know that people who are congenitally blind, their brain builds a picture of the world based on completely different information than most of us take for granted because we're such highly visual species. And so every animal has its own sort of brain, and all of these different brains are the result of ruthless rules of evolutionary selection that have acted over millennia and that have formed and shaped that nervous system so that it processes just the kind of information that that species has to, and basically then ignores things which are not important. So how do we understand our particular form of the human brain? Unlike all of these other animals, we started by our ancestors leaving footprints in the earth, and now we are leaving footprints on the moon. And so how is it that we got to be so different than these other species in terms of these capabilities? And so that's really what we're trying to get at when we study this problem of human brain evolution. So just to outline what I'll be talking about, this is a problem that there's no threat that we'll have it worked out anytime soon, but I'll try to give you a sense of how we think about the problem scientifically, and then a few examples of how we've studied the problem, a couple of examples of stories that, where the evidence seems to be fairly strong of having found particular genes or genetic changes that seem to have had roles in evolution. And then, as I said, I'll close by talking a little bit about the goals of the Allen Discovery Center and how we hope to really catalyze much more rapid discovery in this area in the years ahead. So again, I'm not an anthropologist, but I do want to just orient everyone relatively briefly by reminding people of the sort of relatively recent evolution of human ancestors over the last couple of million years, and remind you that we separated from chimps, our closest living relatives, something like 6 million years ago. And then early hominins went through a series of various shapes and sizes that, like I say, I'm not capable of discussing in any great detail. I will focus on, I'll mention 3 Homo sapiens, of course, us, as well as Neanderthals, and then a third relatively recent but extinct human forebear, the Denisovans, which does not have a skull shown here because, in fact, Denisovans is an extinct human forebearer, but we actually only know about their existence from their DNA. We have a complete DNA sequence of their genome, but no one has yet identified a bone or a skull, which is unambiguously Denisovan. So we know they were there, but we just don't yet know exactly what their form was. So again, just to sort of give a brief cartoonish view of relatively recent evolution to orient people, Homo erectus lived about 1 million to 2 million years ago in Africa, and then Homo erectus spread out of Africa into Europe. Homo erectus gave rise to Neanderthals, Denisovans, and modern humans, all of this happening in Africa. And then Neanderthals and Denisovans actually ruled the entire Eurasian continent for about 150,000 years. That's 3 times as long as modern humans have ruled the Eurasian continent. So in fact, they've been around much longer than we have. For all we know, much longer than we will be. And then modern humans, as a species, Homo sapiens as we recognize them, again, first came into being in Africa at least 200,000 years ago. Best estimates are about 300,000 years ago. And then from Africa, spread into Eurasia, where they met. And actually, there's strong evidence that they interbred with Neanderthals and Denisovans. You may have heard that Europeans all carry about 2 or 3% of our DNA is derived from Neanderthals. Asians carry a certain amount of Denisovan DNA based on this ancient interbreeding between these ancestors. But eventually, modern humans displaced Neanderthals and Denisovans. We don't exactly know how. These numbers refer to the numbers of thousands of years ago that humans hit the Arabian Peninsula or hit Asia or crossed the Bering Strait into North America and eventually colonized the entire world. So the general question is then, how does evolution work on the brain? And this turns out to be, just the question itself turns out to be kind of a complicated one. Of course, it goes without saying that DNA is the canvas of evolution. It is the canvas on which evolution is recorded and then is packaged and transmitted to the next generation. And so that part goes without saying. Comparing DNA sequences between different species then, you can identify parts of the genome or particular sequences that might be specific to each species. And so we can then identify parts of the genome that are specific to humans, and I'll tell you about some of them. And evolution occurs by natural selection, of course, and natural selection then chooses the genomes that are more fit. So basically, ultimately, evolution of the brain must ultimately reflect changes in DNA sequences that regulate the way the brain develops or the way the brain is formed or the way it functions. But the difficulty comes in trying to understand exactly how DNA defines our behavior when it comes to the brain. Because the purpose of the brain is, in fact, to dynamically change based on events. Hopefully, when you leave this room, your brain will have been permanently changed by some little thing that I said or somebody else said. And so, in fact, the human brain, what I just told you is that once humans emerged from Africa, they basically filled every single environment in the entire planet. And so they were endowed with a brain that makes them immensely plastic. And as far as we know, the genome didn't necessarily change a great deal. And so these changes, this plasticity, this learning, is inscribed not in our DNA, but in fact in changes in the neuronal connections in our brains between brain cells. These connections are called synapses, and the neurons between them then change ever so slightly. And so our genome in some way defines us, but in fact it doesn't make us. There's this dynamic paradox between this genetic determinism and this tremendous neuronal flexibility that in fact is is part and parcel of being human. So just to kind of review, this is a picture of a couple of brain cells, and basically what they do is they communicate electrically. They fire these things called action potentials that go down one neuron, and then, in fact, signals are transmitted from one neuron to the next by chemicals, chemical neurotransmitters. And then the brain learns by virtue of the fact that when the activity in one neuron is correlated with the activity of another neuron, then the connections between them become stronger. And so these synapses then have the ability to change or be modified based on patterns of activity, because those patterns of activity reflect what we're exposed to and what we consider important. And so you would think that a lot of the genes that might have changed to make humans so special might have to do with some of these genes that encode proteins that make up parts of the synapses, like these things, these receptors that here are connecting the one neuron shown here, the presynaptic neuron, to the postsynaptic neuron, or perhaps some of these signaling molecules. And here's the names of some of these molecules that are in the neurons themselves. So the other thing is that then the origin story of how evolution worked on our genome to make us human also has to interact with another origin story, which is the story of how an embryonic epithelium prenatally becomes transformed into this neural network that produces intelligence, creativity, social behavior, and the scientific method. And really, this is where I got my start as a developmental neuroscientist and neurologist, is studying abnormalities of this development. So this shows a cross-section of a developing brain of a laboratory animal. This happens to be a ferret, which I'll tell you a little bit more about, a little carnivorous animal that likes to chase mice and chase them into holes and eat them. And so this is just an example of what a mammal looks like during the time when the brain is being put together. And you see these very long little fibers. These are called radial glial fibers. These are the stem cells that put the brain together. This is a cartoon of what it looks like. There are these very primitive stem cells shown in red, and then these are slightly more mature dividing cells. And then the neurons, after they're formed, they migrate away from where the cell bodies of the progenitor cells are, and the neurons collect here on the outer surface of the brain. So anyway, we have to then understand, how do the genes fit into this pattern of brain development? To get back then to the time course of evolution, there are 3 key phases of human brain evolution. Somewhat more than 200,000 years ago, we see that the size of our human forebears tripled compared to that of the chimpanzee. And this was fairly rapid. The x-axis here is millions of years ago. So this is -1 million, 2 million, 3, 4, 5, 6, and back to 10. And you can see the expansion of the volume of the brain from about 1,500 centimeters, which is where we are today, to about 500 centimeters cubed, which was roughly where chimpanzees would be. So this was a very rapid change in evolutionary terms. And then between 200,000 and 50,000 years ago, we see the development of modern human behavior. 50,000 years to the present, we see then changes in different groups since their separation to different parts of the world. But I think, again, it's a huge question. How exactly do we define when humans became modern? How do we define what aspects, based on the fossil record, the anthropological record, how do we know when a pre-human existed with the sort of capabilities that we think of as characteristic of ours today? And here's an example that illustrates that paradox. This is a cave painting from about 5,000 to 10,000 years ago. This happens to be actually this is a watercolor copy of a cave painting from Zimbabwe. And you see this, highly abstract and apparently simplified pattern of drawing. And so we tend to look at that and we think, well, they weren't very good artists back then, were they? But then, of course, if you see these by Giacometti, you say, well, this is obviously a deliberate abstraction created for artistic effect. And so the question is, what do we really make of these cave paintings? And to what extent are they as primitive as we might be easily to interpret? And the reality actually is that Giacometti was in fact influenced by those cave paintings to develop his unique style, because in fact those cave paintings were described just when Giacometti was in his formative years. Okay, so next I'll tell you about some of the sorts of things that can happen to the genome that can underlie evolution. And basically we think of evolution as occurring by 3 general mechanisms in the genome. There's only so many things that can happen to a genome. And so the simplest thing to conceptualize is that you can add a new gene to the genome. So this is a picture of a genome of a single cell. These are the chromosomes, the 46 chromosomes that make up our genetic material. Each of them, when you stain them like this, they have little stripes. These are the 2 chromosome 1s, which are named chromosome 1 because they're the largest. And then you see that the chromosomes are numbered in descending order of size. So you can then take a chromosome here, take a closer look at it, and you can go on a website to see what your genome actually looks like. So this is a schematic of a chromosome here. You can see those same black and white stripes that highlight the chromosome. The red box is highlighting this particular segment of the genome that then is shown at higher magnification. And then these blue things are actually genes. They each have like little names, typically 3 or 4 letters or something like that. Here's one gene, here's another gene, here's another gene. There's spaces where there's no gene in there. And so the simplest thing you can imagine is that you just add a new gene to the genome. Now this turns out to be a relatively rare event because there aren't just genes sitting around on the table that nature can sort of plug in. Genes generally have to come from somewhere, and they're complicated things. They have to be so precisely organized so that they encode a protein. But so the more common way in which gene addition will happen is actually by the duplication of an existing gene and then tweaking it a little bit so that it does something slightly different. And I'll show you an example of that. When you see duplications of a part of the genome or a loss of the part of the genome, we call them copy number variants because it's a change in the copy number of the genome. So this is what a normal chromosome would look like, and I've just highlighted 4 segments of the genome labeled A, B, C, and D. You can have a deletion that removes, so these are the 2 homologous pairs of the same chromosome, right? We have 2 copies of each chromosome. You have 2 copies of chromosome 2, 2 copies of chromosome 1. In this case, one of those 2 copies has lost the segment C. Or you can have an insertion where segment C is now duplicated. And so what this does is it duplicates all of the genes in that segment. And that segment might have 10 genes in it. It might have 30 genes in it. It might be a segment similar to the one that I just showed you. And so here's a segment of the genome in the human genome that happens to be a particularly fragile place. And it's subject to frequent duplication, actually, as a cause of medical disease. And so this is chromosome 1 laid on its side. And there's this segment of it here highlighted, chromosome 1q21.1. And this actually is blown up then. And you can see there's several copies of this gene called Notch2N. There's an A and a B and a C copy. All of these genes were originally derived from an initial forebear gene called Notch2. Notch just is we have to give genes fancy names. Notch happens to be named after a fly because the gene was first found in the fly, and when it's mutated, it causes a notch in the fly's wing, which is how it was first recognized a century ago. And then we found out that all species have a notch gene, or virtually all eukaryotic species have not eukaryotic, but vertebrates and invertebrates. Most animals and flies have a notch gene. So humans actually have several. There was Notch2, and then that got duplicated several times over. This particular part of the genome, actually, though, in humans, tends to vary a lot. And in fact, you can have a deletion of this large segment here, and that causes a disease. That causes that is associated with a higher risk of schizophrenia and also makes the brain small. And then other patients can actually have the corresponding duplication of that same region. And the duplication is associated with a big brain, but also predisposes to autism spectrum disorders. And it's a whole other talk to tell you how that might happen. But there are these reciprocal deletions and duplications suggesting that this part of the genome actually might have something in it that's regulating the size of the brain. And as I just told you, the size of the brain is very important in recent human evolution. And this just shows what microcephaly looks like. Micro means small. Cephaly just means brain. So this is a small brain. This is a big brain. And you can see that in this case, the brain is smaller than this normal here, but otherwise looks fairly normally patterned. It has all the usual folds that we call gyri and sulci, which are just fancy Greek words for hills and valleys that characterize the outer surface of the brain. And you can see over here, this is the circumference measured around the head of kids. And kids that have deletions, their head circumference is small. Kids that have the duplication, their head size is enlarged. And so this is a part of the genome that was recognized medically as regulating the size of the brain. It also contained this brain notch, which had been known to regulate stem cells in many different parts of the body. The wing, as well as the brain, because it turns out notch is expressed in many different tissues. And so recently, 2 groups, one in Seattle led by Evan Eichler and another group in Europe, actually started looking more carefully at notch and saying, well, hey, you know, the size of the brain is important in evolution. Might this notch gene be part of that? And so they studied the evolutionary structure of this Notch2 area. And it turns out that originally, orangutans and other great apes only have a single copy of Notch2. And then in the lineage leading to human, it duplicated to create these Notch2NL things. And then, in fact, on the human lineage, there were further duplications that created Notch2NL versions A, B, and C. So in fact, humans have multiple different copies of Notch as their brain got bigger and bigger. Whereas, again, the great apes with smaller brains have fewer copies of this. And in fact, the same group did a series of studies to show that this Notch2 NL regulates the stem cells in the brain and actually then helps tell them to keep proliferating, to make a bigger brain instead of to stop proliferating, which is what you have to do to make neurons. So now that I've given you a course on genetics and evolution, I'll give you a short course on neuroscience, tell you a little bit about how stem cells in the brain make neurons. And so this is then a schematic of a fetus again, similar to that picture that I showed you that shows these stem cells, which are along the ventricle, which is a hollow area deep in the brain. And then the cerebral cortex develops from the lining of that hollow tube. And again, the stem cells are these elongated things on the inner surface. And they give rise then, they have these elongated processes. This movie runs a little slow, but the Notch2 gene is expressed in these progenitor cells. Progenitor cells are giving rise to these bubbles, which are representative of postmitotic neurons. Neurons in your brain don't divide. And they're not like your skin where they'd regenerate if you get a cut. And so the number of neurons that you have when you're born, with a few exceptions in a couple of parts of the brain, are basically the same number of neurons that you're always going to have. And so these neurons are born prenatally during the first couple of trimesters of fetal development. And they become postmitotic. They migrate to the outer surface of the brain. And as you collect more and more of them, the cortex gets understandably larger. So the Notch gene then is expressed in these cells, not in the neurons, but in the cells that give rise to the neurons. And it's basically a switch that tells those stem cells. Stem cells are great for making the brain, but you can't think with your stem cells. You only think with your neurons. The neurons are the things that have synapses. So at some point, if you're going to be smart, the stem cells have to stop dividing and give you these neurons that can then develop these sophisticated connections and synapses. So Notch then seems to work in these stem cells, and this is another realistic picture, again from ferrets using a bunch of different colors. The stem cells are down here, shown schematically in this cartoon. And they give rise to the neurons that migrate up here. They stop migrating and they make their connections. And so it seems then that this Notch, this increase in the number of copies of Notch, seems to be able to then regulate the size of the brain evolutionarily as well as medically. And so then it seems like humans have just the right number of copies of this Notch2, because if we have too many, we have this problem with the brain being too big, and that causes cognitive problems. And if we have not enough, that causes problems as well. So a somewhat more common mechanism of evolution is to take a gene which is already in there and tweak it a little bit by changing the sequence of the gene. Now, as you know, DNA encodes RNA, then the RNA codes for a particular sequence of amino acids that generates a protein. And so the biochemical patterns of a protein are defined by its amino acid sequence. If you swap one amino acid for another, it can change the shape of the protein and hence its biochemistry. So this is, again, an important mechanism of evolution. And again, one of the best characterized examples of this is a gene that regulates the size of the brain. So in this case, again, we have a gene here. You blow it up, and we can look more carefully in detail at the sequence. Here, the amino acids are each abbreviated by a single letter. And then if you introduce a few changes in that amino acid sequence, that can then change, as I say, the biochemistry of the protein in a way. And so then, in this case, for evolution, nature doesn't have to invent a completely new gene, but it can dow an existing gene with new properties. And so again, we get back to this rapid change in brain size in relatively recent evolution. And this is actually where I stumbled into studying evolution now more than 10 years ago, when we worked with Jeff Woods to identify one of the first genes which, when completely disabled in humans, causes microcephaly. So this is just a single gene that regulates the size of the brain. This gene is called ASPM. Again, named after a fly gene, abnormal spindle. ASP is the fly gene, and M, we renamed it ASPM for abnormal spindle microcephaly. And so when we first cloned this gene, we noticed a bizarre thing about it, that the actual size of the encoded protein was way larger in humans than it was in fruit flies, where it had first where ASP had first been identified. And then the size in the mouse was sort of in between. And in fact, the size in the worm was even tinier than in the fruit fly. So it looked like the actual encoded protein got bigger and bigger over the course of evolution. And so we wanted to then test the idea that it might actually be a target of evolutionary selection. And you can't really do that between widely divergent species, and so we did it between more closely related species, just among the great apes. And there's a statistical test you can apply to this that looks at how common it is that an amino acid gets substituted. Because most of the time, evolution has created genes exactly the way evolution wants those genes to be. And any change in the sequence of the gene is usually trouble. It's usually a mutation and typically causes disease. And that's, in fact, the sorts of mutations that were causing this. But every once in a blue moon, a little tweak in the sequence of a protein might not be damaging and might improve it in some way. But this is much less common than for a change to be damaging. So for a normal protein, the sequence of that protein is carefully guarded, and so we can look at changes in the DNA sequence, either that change the sequence of the protein or that don't change the sequence of the protein. For a normal gene, the vast majority of changes in the DNA conserve the protein sequence, but for this, and so we see a ratio of those two that's less than 1. For this particular gene, there's an excess, a ratio greater than 1 of changes that actually alter the sequence of the protein. And this is quite a rare event, providing strong evidence that, in fact, this gene was changed in some way, pari passu with the increase of the size of the brain in recent evolutionary time. Now, the first thing you want to do as a biologist when you identify a gene like this, you say, wow, this is the mechanism of evolution. Let's try to get at the cellular biological mechanism of how this ASP thing works. And so what we did was we created a gene mutation in mice to try to model what the gene might be doing in humans. And here you see the comparison between a normal human and a human that has an ASPM mutation. Again, the brain is reduced up to 2/3 of its size. A very small brain, but normally patterned. Well, you knock it out in mice and almost nothing happens. You can't really tell which are the mutants and which are the wild types. There's about a 10% reduction in the size of the brain. And so this was kind of frustrating. And the point seems to be that, in fact, the gene is doing different things in these different species. Now, when you actually look at the stem cells in the human brain and you compare them to the mice, in fact, even the progenitor cells are very different. It just looks like the mouse brain is being put together in a very different way. The mouse has 2 types of stem cells, the red and the yellow ones. And then Ferret is an example of a more common sort of mammal pattern. They add these green progenitor cells that are long like the red ones, but not quite all the way up and from the very top to the very bottom. In humans, these cells, these green cells called outer radial glial cells, or ORGs, they are the most abundant progenitor of all in the brain. Many of my colleagues, and maybe me a little bit, think that these brains might to a certain extent be the seat of the soul, because they become so abundant in humans. They seem to be doing something extremely important, and yet they are absent in mice. And here you see, actually, so there's this stem cell zone down here has these red type cells, and that's about the same in mice and in ferrets and in humans. In fact, the size of that is maintained about the same, but this outer zone that contains these green cells, in fact, becomes a little expanded in ferrets compared to mice, and then hugely expanded in humans compared to actually any other species. But all mammals, virtually all mammals, are very different than rodents in this way. Now, I told you that, in fact, the mouse and the human have fairly different sequences of this ASPM gene. And this shows a schematic of the structure of the ASPM protein between humans and ferrets and mice. And as I told you, the mice has just a shorter encoded protein. And the majority of this protein is encoded by these repeated domains, which are called IQ domains or IQ repeats. And they're just called IQ because every one of them starts with an isoleucine, which is abbreviated with an I, that's an amino acid, and glutamine, which is a different amino acid abbreviated Q. But when we cloned this gene, Jeff Woods made the joke that this is how we know that God has a sense of humor, because could it actually be possible that the number of IQ domains specifies your IQ? Could it actually be that easy, that evolution had it that way? But anyway, clearly, not only are the stem cells different in mice and humans, but also the structure of this gene. And if you actually look at the relative conservation of the ASPM gene, here it shows between humans and chimps and gorillas, you can see that, in fact, rodents are a real outlier with a very low level of similarity, suggesting the gene is most different between rodents compared to others. Other mammalian species. And so what we wanted to do is, again, to try to get at the cellular biological mechanism of this gene, we wanted to create an animal model that was more close to the human. And as I mentioned, the Drosophila has only 21 of these repeats. And so actually, most of the loss of these repeats in the mouse is due to a deletion of just one little spot of the genome, in some ways a single evolutionary event that actually removes many of these IQ repeats. So originally we thought, in fact, that the mouse version was kind of like the ancestral version to the human, that the human gene grew. It turns out, actually, that the human has the conventional version for mammals, and that mice actually seem to have gone out of their way to shorten the protein, to make a smaller encoded protein, that is actually the evolutionary event that's actually relatively unique to rodents. And so for this reason, then, we created an animal model in these ferrets, because these days you can knock out a gene in anything. You've heard about CRISPR and all this stuff. And so we thought, well, we'll just make a large animal model. It happens that ferrets are convenient to work with. They breed very easily in captivity. They have huge litters. They're very easy to study developmentally. So we targeted it in ferrets. And here you can see that they make a much better model compared to humans. The brain is reduced to by about 40% of its size. In fact, it has a defect much like the humans. What actually happens in the human brain when you lose ASPM is that the cortex doesn't become thinner, it becomes smaller in surface area. So the thickness is normal, about 100%, but the area is smaller, so that the cortex is reduced in size. And the same exact thing happens in the ferret, where in fact the surface area is greatly reduced in size, but the thickness is barely reduced in size at all. And in fact, it's an amazing thing about this ferret that the very parts of the brain that are thought to be most recently evolved, like the frontal cortex and the corpus callosum and the lateral cortex, are those that are most severely affected by the mutation. And then parts of the brain that we think of as very ancestral, that are shared by reptiles and so forth, are barely affected by this mutation. And so the mutation actually causes a fairly simple change in the cortex that I'll just show you about. This is a section of a newborn ferret, which is in the middle of the process of making its neurons. So those stem cells are down here, the neurons are collecting up here, and so the stem cells are illustrated here in color. The neurons are collecting up here, but you can't see them. And what happens when you lack ASPM is that you have a shift of these stem cells to these guys. You see that they all collect in this big clump, which you don't see in the normal. And these outer radial glial cells, or ORGs, are formed precociously. And so it appears that then ASPM is also a slightly different kind of switch that controls this change from this kind of stem cell to a slightly more differentiated or mature stem cell that has less in the way of proliferative properties. And so the loss of both species, it reduces the cortical surface area without really changing the cortical thickness. We think that basically these guys, the red ones, control the surface area. They divide and they end up next to each other and next to each other, and they expand the surface area of the cortex. And then these guys, once they leave the ventricle, in fact, they generate the different layers of the cortex above. And so there's a sequence of development. And ASPM affects this step and that step mainly. And so it does indeed, so this is still a hypothesis that tweaking ASPM, changing it in subtle ways, might regulate the size of the brain, but it certainly, all of the biology points in that direction. But then also it suggests this thing about mice. Again, we think of mice as the prototypical lab model for humans, and that they're sort of a proto-mammal. But in fact, now I think of mice as exquisitely selected by evolution, that in fact, the size of the brain for a mouse works perfectly for a mouse, because mice are ruthlessly evolved to be able to crawl into tiny holes that a ferret can't fit into. And so they want to have a small head because it's the size of the head that actually determines the size of the hole you can crawl into. And in fact, if their heads were any bigger, then they would be eaten. Or it's something like that. I mean, perhaps that's a bit simplistic, but in fact, mice, it seems that mice don't want to have a smaller brain because, again, the entire rodent order has disrupted to ASPM to a certain extent. So now, the last kind of genomic change that underlies evolution is probably the most common, but is the hardest to study. And this is changes to the dark matter of the genome, the non-coding parts of the genome. And so this is a part of the gene where the genome where there's no genes there. It just seems like it's a spot in the middle of nowhere. And what is in the dark matter of the genome? Well, some of it is true junk, but a lot of it are on-off switches, are regulators that tell the genes near them, in fact, where and when to be expressed, and so that they control the patterns or the levels of expression of nearby genes. And so I'll just tell you a little bit about how we're trying to get at the function of some of these non-coding parts of the genome. And we've been focusing on a particular subset of these non-coding parts of the genome that have been already identified by a variety of different laboratories, and they're referred to as human accelerated regions. And accelerated just means they show accelerated change in the branch of evolution that leads to humans. And so this is an example of the genome of a mouse, a ferret, a rhesus, a gorilla, sort of lined up and compared. And a part of the genome that is neutral, your average genome, will have a certain number of substitutions if you compare one species to another. And the human will have about the same number of substitutions relative to any other given species as anyone else to one another. Now, there are other parts of the genome that are intensely conserved, that are identical or almost always identical or almost completely identical in a large number of different mammals. And when you compare genomes and you see this conservation, this is nature's way of telling you that a sequence is really, really important and that it does not tolerate any sort of change and that any change is very likely to be damaging. And so you can identify these sequences of the genome as having essential functions just by virtue of the fact that they're so highly conserved between species. And then there are the human accelerated regions. Human accelerated regions are defined as parts of the genome that are highly conserved in many, many different species, but then are a little different in humans compared to other species. And so this conservation between many species suggests that they have essential functions, and then the changes between humans and other species can have a variety of explanations, but one of those explanations might be that that essential function has changed just a little bit between humans and other species. It can also mean, of course, that in fact an essential function has lost its essential function, has become garbage, and so that's why all of these things have been introduced. So we need additional information to prove which of the human accelerated regions are really important by trying to explore what the actual essential functions of these HARS are. Now, we know that for many of these HARS, they've been identified by a variety of experiments as actually being sequences that direct patterns of gene expression. And in some cases, you can even identify HARS that, where the human version of the HAR directs a different pattern of gene expression than the chimp version of the HAR. And here's an example. This is HAR number 5, and the chimp version, which PT stands for chimp, pan-troglodytes, that drives expression, but at a relatively low level in this little blue dot in this embryonic mouse, and this happens to correspond to the embryonic cerebral cortex in that developing mouse. And then if you take the human version, you can see that the expression is really, really juiced up in the cerebral cortex and spreads to other parts of the back part of the brain and the spinal cord. And so in fact, the evolutionary changes in the sequence of this particular HAR seem to confer a change in function. The problem is there are more than 3,000 spots in the genome that have been identified as HARS. Each one of these HARS is about 100 to 300 base pairs long. Sometimes they're in genes. Most of the time, they're in the middle of nowhere, and we don't know that much about what they're connected to. And so the challenge is to figure out whether all 3,000 of these are important to making us human, or whether some of them are more important than others. And so we wanted to ask the question whether we can identify HARS that have essential functions. We thought, well, okay, humans have social structures that look a little different than chimpanzees. And so there must be sequences in the genome that regulate those changes in social structure. And so if HARS are important in defining us as human, then some of them should have essential neurological functions. So if we look at patients that have a very severe disruption of a HAR, maybe they have a loss of function that might tell us about what that particular HAR is doing. And by identifying those HARs that have essential genetic functions in humans, we can then winnow down this list of 3,000 HARs to try to identify those that might be most important. And so a couple of years ago, we showed that indeed some of the HARs can be disrupted in kids that have autism or intellectual disability. This is a particular HAR number 426. And we found 2 different families that had autism and intellectual disability where they in fact had disruptions of this HAR 426. And HAR 426 we found out connects to a gene which is called CUX1. CUX is similar to another fly gene called CUT. And CUX and its related gene CUT, they have remarkable, they also are sort of switches, but they don't control stem cells, they control neurons. So that in flies, if you lose CUT, you take a complicated looking neuron and it gets simpler, or if you get too much CUT, it takes a simple looking neuron and makes it more complicated. So in fact, CUT controls the elaboration of the patterns of connectivity of neurons. And the same thing happens to be true, Same thing is true of the mouse version, that if you lose it, you take a complicated neuron and you make them simpler. If you overexpress it, you take a simpler neuron and you make it more complicated. So again, nature seems to have very carefully titrated the exact right amount of CUT for the shape and size of the neurons that it wants. And in fact, interestingly, CUT is expressed only in the upper layers of the cortex, layers 2, 3, and 4. These happen to be the layers that are most highly elaborated in humans and which are very simple and in rodents and even missing in some of the smallest mammals like hedgehogs. So human brain evolution then is likely a tinkering at many different sites in the genome. This is an idea first developed by Francois Jacob now 40 years ago. There are probably many sites in the genome. There are probably many mechanisms, but a finite number of mechanisms. It's a big problem, but it does seem to be a finite problem because the genome is, after all, only 6 billion base pairs. So it's non-coding as well as coding gene evolution is involved. As I say, at this point, we have too many candidates for sites that might be involved. The key is to narrow them down to which ones are most important. What proportion of these are essential to the brain and what proportion have evolutionary roles. So I'm running out of time, but I still have a couple of minutes to just briefly outline what we're trying to do with the center. One thing we're trying to do is create the world's largest resource of DNA sequence of ancient and archaic humans. This is the work of David Reich, and he'll be coming to speak to you soon. And this sequence allows us to do more kinds of analyses to identify these human-specific changes, or actually to find some of the sequences changes that people have claimed are human-specific and find that actually maybe they're not so important. And then we also want to study these aspects of learning and memory that involve changes in gene transcription to try to understand some of these that might be evolutionarily dynamic. We feel that we can't help but learn about genetic brain diseases of children as a part of doing this work, because not only is it important to identify the causes of childhood brain disease, but this is in fact how we identify the sequences in the genome that are essential. Just as a biologist, if you want to understand what's essential in the human genome, you can only do that in the case of humans by studying disease, because that really tells you what matters in the genome. So, like I said, David is going to be talking about large-scale sequencing of these old bones that get dug up that are 5, 10,000, 50 years old. And he's developed ways of purifying the DNA, the very small amounts of DNA, and sequencing it to create the largest resource of ancient DNA. And in fact, David's lab has generated more than half of the world's collection of these ancient genomes. And so having this larger DNA sequence resource, capturing more of the diversity of humans, we can capture not only the diversity of living humans, but we can capture the diversity of humans which are no longer living. We can get the broadest sense of what sort of range of the genome is consistent with being a normal human. We can then identify the various changes that occur at each stage of the evolutionary tree. And then, as I said, we also want to identify physiologically in the genome what are the parts of the genome that regulate aspects of learning and memory and this control of gene transcription that I told you about that's so important. The control, for example, of the levels of that cut gene. And that's where Mike Greenberg is taking the lead, because this has been the goal, the mission of his lab for many years. And here's an example. He found a gene called osteochrin, which is expressed in the bone in the mouse. That's why it's called osteochrin. But in fact, in the humans, it's actually expressed very highly in neurons in relationship to their activity. So when neurons are active or depolarized, their levels of expression of osteochrin go way up in humans. And in fact, if you take a monkey and you close one eye, the cells that are responding to the open eye express high levels of osteochrin, and the cells that are not active because they're exposed to the closed eye, in fact, show low levels. But this is a gene that has totally changed its function between mice and humans. It's not a bone gene anymore in humans, it's a brain gene. And so this is another way of identifying genes that might have dynamic evolutionary effects. And then, like I say, we'll use the genetics of pediatric brain disease to parse out which of these evolutionarily dynamic sequences actually have essential functions. And we have to still do animal models, but we'll need some new animal models, things like not just mice, but ferrets, or even potentially other animal species as well. So. Thank you very much for your attention. I'm just going to acknowledge some of the people. The ASPM work in my lab was done by two postdocs, actually now both former postdocs. The HAR mutation work was done by Ryan Doan, a postdoctoral fellow in the lab. And there are many other people that contributed that I don't have time to acknowledge all of them, since this is intended as a sort of a general overview talk. But again, thanks very much for your attention. I'd be happy to answer questions.